By definition, cannabinoids are the terpene phenolic constituents of cannabis sativa L. There are three types of cannabinoids. First are endocannabinoids that occur naturally in your brain, named 2-arachidonoglycerol and anandamide. The second group are called phytocannabinoids. They are plant-derived natural compounds sharing chemical similarity with cannabinoids and or being capable of directly interacting with cannabinoid receptors. And the third type are synthetic cannabinoids that are created in laboratories and are not known to exist naturally. There are over 100 identified cannabinoids in the cannabis plant. 2 are cannabidiol, or CBD for short, and cannabigerol, or we call it CBG. As for CBD and CBG's history, the first person who was successful in extracting CBD from the cannabis plant was a chemist who graduated from Harvard University named Roger Adams, but he didn't even know what he did. It wasn't until a few years later, in 1946, when a man by the name of Dr. Walter S. Lowe, he conducted the first CBD test on lab animals. These tests gave proof that CBD does not cause an altered mental state, and that was good news. Now, that same year, over in Israel, Dr. Raphael Mechoulam, he identified CBD's three-dimensional structure, and that's why he's often credited as the scientist who discovered CBD. Now, further research continued in the 60s on primates, and finally, British pharmacologists released the first CBD oil meant for therapeutic use. In the next few decades, the research continued, and then in 1980, Dr. Mechoulam made another breakthrough in CBD history when he ran a study which showed how CBD, cannabidiol, could be a key factor in treating epilepsy. Doctors, health practitioners all over the world are now understanding the wide benefits of CBD and CBG and are recommending it for therapeutic use for their patients. Cannabigerol, or CBG, was first discovered in 1964 and is known as the mother of all cannabinoids because in its acidic form is the chemical precursor or foundation to CBD, THC, and another cannabinoid called CBC. By the end of the harvest, the cannabis harvest for the plant, a group of enzymes break down the acidic form of CBG to produce the cannabinoids THC, CBD, and CBC. So any leftover CBGA can become decarboxylated and turned into CBG. Now, CBG and its intoxicating counterpart, THC, are found in high concentrations in these mature cannabis plants. But CBG, on the other hand, is found in trace quantities. In fact, less than 1% by dry weight. So most of us in the field have nicknamed it the Rolls-Royce cannabinoid because it takes thousands of pounds of biomass to produce small amounts of CBG, and that gets expensive.